very happy for them to invite me for their symposium, and I will try to do my best to help their programs as much as I can. So, when Hank said, do you mind getting together and sharing some of your insights? And I decided that, maybe I should begin to tell you who I am, as a person, as a scientist, how my journey started and where am I in my journey. And then maybe talk a little bit about why the genetics of HSP and PLS is important. And then looking towards the future, like what are we going to do to build effective treatment strategies, and what are the limiting factors, and what is the path forward, and of course with the hope that it's going to generate some discussions and that we can talk together. So for who I am, so as you know, my name is Hande Ozdinler. So it's not like Jack, Joe, Jill. So I was not born in the United States. I was born in Istanbul, Turkey. So and I did not have, when I was a teenager, I did not have any intentions of coming to United States, because I had a very happy family. We had a nice life in Istanbul. It was never my intention to come to United States at the time. And I went to Boğaziçi University, and this is actually my campus. This photograph is my campus, and this is the Bosphorus Bridge. This is the Bosphorus Bridge, the second bridge. And these are the buildings of my campus. And, this is where I used to hang out as an undergrad student. As you can imagine, it was beautiful, very, very nice campus. And this was actually, is an American university built in Turkey. And the language of education is English. And for the university exam, they select the top 1% of students. So it was one of the best universities, and I was very happy with my school. And I graduated from molecular biology and genetics department. Then for my master's, I did biotechnology master's with the combination of molecular biology, genetics, and chemical engineering. And that was a very new program. And I was actually the first person to clone a gene from Thermus aquaticus. That's the bacteria that can live in very high temperatures. And that bacteria actually came from the state park here in the United States. That bacteria lives in the boiling waters. So we were able to clone genes, and imagine that I cloned a gene when there was no PCR machine available at the time. So that was the first in Turkey at the time, and we got patents, invention disclosures, blah, blah, blah. I got Best Master Thesis Award. I was a very good student. Then at the time, if you are a very good student in Boğaziçi, the trend was to go to United States for PhD. And so, this was my family. This is me. This is my brother. This is my mother. And so I was a very happy graduate. But then I said, okay, then, I'm going to go to United States and I'm going to study plant biotechnology, because I was cloning genes, right? So I thought, if I can clone the right gene into plants, then we can make sustainable plants, or plants that can even live in environments where there's not enough water, or plants that they don't get sick, or, we can have a solution for the deforestation, or, those were the things I had in my mind. So my boyfriend at the time came to Georgia Tech, Atlanta. So then, what do I know? I just looked at the universities around Atlanta, and there was this university at University of South Carolina. So this is the campus, this is the photograph. So I came to University of South Carolina in Columbia as a PhD student. And, that was a big culture shock for me, because going from Istanbul, 10 million people, and to Columbia, South Carolina, where there's no one on the streets, and it's a very small city. And but I liked it. Something very bad happened though. My brother passed away, unfortunately, at the age of 23, and he had stroke. So he died in my father's arms, and he was very young, and he just died. He had stroke. And I was on my second year for PhD. You know, I have taken all my classes and I was going to study plant biotechnology, and I was in a very good lab. He was a Harvard Hughes professor, and it was very nice. But I had to study the brain because I didn't know anything about the brain, and my brother died because of stroke, and I had to understand the brain, and I knew nothing about the brain. So then, you know, I switched all my program. I switched my university. I started looking for universities who would accept me. And then I came to New Orleans. I came to LSU Health Sciences Center, New Orleans, and I studied neuroscience for the PhD program. And I worked with Reha Erzurumlu. So he was actually graduated from MIT, and he was working on nervous system development, path recognition, and somatosensory cortex mapping, and so forth. So I became a PhD student in his lab, and I wanted to be in the MD PhD program and learn as much as possible. You know, I was like a sponge. I just wanted to learn anything, everything about the brain and the neuroscience, and I just wanted. It's as if like if I learned all I can, it's like it was just like my service to my brother. I don't know how to explain it to you, but I had to learn it, you know. So I was in the, and you know, I was in my PhD program, and then I said, you know, I want to do a PhD in cell biology, anatomy, neuroscience, like, you know, in 3 different topics. They said, we're not going to call you PhD, like you're just going to be PhD. I said, that's fine. I just want to learn as much as possible. So everybody had to publish 1 paper. I had to publish 3 papers. Everybody had to do, you know, 3 committee members. I had 9 committee members. Just, I made it so hard for myself. And I had 6 papers published when I was a PhD student. I was a stellar student. And when I graduated from PhD, I went to Harvard Medical School for postdoc. And I was very lucky because Dr. Jeff Mackless accepted me to the neurosurgery program. And I also worked together with Dr. Brown, and I became one of the recipients of Harvard's Center for Nervous System Repair Fellowship. So I became a Harvard Fellow. And I worked so hard for 6 years. And some of my experiments didn't work. I kept working. And so it was a very productive postdoctoral experience for me. And I had excellent mentors. Then I moved to Northwestern because at the time, Jack Kessler was the department chair, and he said, Hande, we want to build the second Les Turner lab, and we would like to hire someone who would like to be the founding director of the second Les Turner lab. And, you know, they had so many people interviewing and looking for the position and so forth. And I was accepted, and I was about 35 or 36 at the time, and I became the founding director of the second Les Turner lab. And it was very important for me because I received a huge support from the first board of Les Turner, and our lab became the first upper motor neuron lab, one of the few maybe in the world. And we focused on understanding why the upper motor neurons degenerate, because these neurons are important for ALS, for HSP, for PLS, and also for spinal cord injury patients. And at the time, really, there weren't really tools or anything to understand their biology or their pathology. So, you know, I've been there for about more than 11 years now. I'm an associate professor. We have the Ozdinler lab, the upper motor neuron lab. And so, you know, I'm a faculty member at the Chemistry of Life Processes Institute, the Mesolem Center for Cognitive Neurology and Alzheimer's Disease, as well as the Robert H. Lurie Comprehensive Cancer Center, because to understand the biology of complex diseases, we really have to form collaborations, and I'm going to talk to you more about this. So in our lab, you know, it's been maybe 10, 11 years, and we became the first lab dedicated to understand the biology of upper motor neurons. So it was just a pure, we focus on the biology and pathology of upper motor neurons. Then we made the first reporter line for upper motor neurons. We made disease models where upper motor neurons are fluorescent, and we developed drug discovery platform that utilizes upper motor neuron health as a readout. And you can imagine how important this is, because there are no drugs for upper motor neurons, and for preclinical assays, you really need to understand if the compound of interest also improves the health of the upper motor neurons. So we also began to reveal the underlying causes of upper motor neuron degeneration, and you will find that it is very heterogeneous. Even though patients may display a similar clinical outcome, the underlying causes may be different. And if that's the case, one drug is not going to cure everyone. So we really have to understand what is the mechanism and what the treatment strategy should be for that mechanism. And then, of course, we began to develop cell-based treatment strategies for upper motor neurons. And also, we are identifying biomarkers that inform on the timing and extent of upper motor neuron loss in patients. And you can imagine that all this is an amazing achievement in such a short time. And I think we were able to achieve this because we were very focused. We were focused on the cell biology of upper motor neurons, and we wanted to understand their biology and pathology at the same time. And as you understand them more, you also begin to reveal therapies or approaches for therapies, right? And so for the second part, you remember I said why the HSP and PLS genes are important. So here, we are actually invited to write a review. It's going to come up soon, and this table will be on that review. And there are so many genes identified to be either linked or associated with HSP and PLS. And of course, HSP and PLS may be clinically different or considered different diseases, right? But at the end of the day, it is the upper motor neurons that die in patients. And one of them has more genetic background, and the other one maybe is more sporadic. And so when things are sporadic, it does not mean that there is no genetic cause. It may mean that we don't know the genetics yet. And you can see here, there are about 96 genes that are linked or associated. So when I say linked, I mean when there is a mutation, you get the disease. So there's like a parallel, like you have the mutation, you have the disease, right? Associated means that when you have mutation or when there's some problems with the genetics of that gene, the chance for you to have HSP or PLS increases. So there's not really a one-to-one correlation, but it increases the chance for you to have HSP or PLS. And you may see that some of them are highlighted as yellow because we were writing a review about importance of lipids for upper motor neurons. And these are the genes that are important for the lipid homeostasis, maintaining of lipid homeostasis, and the importance of lipids for upper motor neurons. So there is a wide variety of genes here, right? And this is the question of our time. And that is, even though these genes all code for different proteins, okay? How come when a patient has a mutation in one of them, in any one of them, develop a similar clinical phenotype? So there must be some converging paths, which may explain the divergence of the disease. So knowing which mutation you have or what is the underlying genetic cause is extremely important, because then you may begin to reveal what is the cellular event that cannot be performed in the presence of that mutation or of that problem. So we actually did a very similar study for ALS, and we published it before. And we think that every action that takes place in a cell, in a neuron, is mediated by mainly by protein-protein interactions, because proteins talk to each other, because they connect with each other, right? This creates a cellular outcome, which we call, let's say, the canonical pathways. So there are, for some canonical pathways, for some cellular events, there are some proteins that are that play very important role. So when there is a mutation in one gene, that means the protein coded by that gene cannot perform its function. So imagine this. When there is a mutation in one gene, that gene codes for a protein. And when there is a mutation, that means the protein coded by that gene cannot do what it is supposed to do. So this leads to two problems. One is that when that function is required in the cell, there's no protein to do it. Second, the mutated protein or the wrong, mistaken protein, or now it is defective protein, may actually have a toxic function. That mutated protein may aggregate with other proteins. That mutated protein may go somewhere else and destroy other proteins. That mutated protein may become a rebel, may become a angry protein, and it can create damage inside the cell. So you have two problems. One of them is a dangerous protein inside the cell, or production of a dangerous protein inside the cell. And the second is the lack of the protein that you require. So then the cell becomes under stress, like, I need this now, I need this now. But now the protein says, I don't care, I don't care, I'm just going to walk around and destroy everyone. So you cannot function like that. And then at first it may say, okay, maybe I will do this, maybe I will do that. And it tries to maintain homeostasis, it tries to maintain balance. But after a while it says, I really need that protein. It says, I don't care, I don't. And so, okay, after a while, the vulnerability turns into neurodegeneration, because first it's vulnerable. It says, I need this. And then it becomes, I can't do this anymore. That's it. I can't do this anymore. And then they raise a red flag and they give up. The moment they give up is the moment neurodegeneration starts. But when we realize what the problem is, we can tell the neuron, oh, don't worry. If that protein doesn't do what it needs supposed to do, we'll give you another one. Here you go. This one is fine. This one is working. Or, Tim is not doing its job. Well, we'll give you Tom, and Tom will do Tim's job. Okay, fine. So we can find a solution to the problem as long as we define the problem, as long as we understand the problem. So by looking at protein-protein interactions, we can actually understand the basis of the problem. So that's what I was trying to tell you. And again, so we can definitely go from mutations to protein networks. And this is the basis of identifying new targets for drug development. And I think we have made very important progress in that field. So here I wanted to bring to your attention, as we build more knowledge, right? Our goal is to cure the disease, and our goal is to help the patients. But there are many, many limiting factors. So one way to look at limiting factors is, how do we turn them into advantage? Because there will be a limiting factor in every field. There's always a limiting factor. So there's no running away from limiting factors. So one way to deal with a limiting factor is to turn them into advantage. And I think that there are many opportunities for us to turn the limiting factors into advantage. For example, they say HSP, PLS, these are rare diseases. Oh my God. So the drug companies are not interested because these are rare diseases and very few patients, and developing medicine for just very few patients is not a good strategy. It's not profitable, it's not worth it. Sometimes they say that, and it's not good. Okay, but this gives us an opportunity to develop personalized medicine. This gives us an opportunity to move the medicine field forward by developing personalized medicine approaches. And what do I mean by that? So maybe in the future, we will not believe that one drug is going to cure all. There will be no one drug that cures it all. And we will not really talk about, Alzheimer's, Parkinson's, ALS, HSP, PLS as diseases, and we will not really have drugs for diseases, but we will have drugs for the underlying causes. So once we have the drugs for the underlying causes, then we don't have to worry about rare diseases, right? We don't have to worry about which disease you have. If we understand that the underlying cause, for example, in a subpopulation of HSP or PLS patients is because, let's say, mitochondria is not functioning properly. That's the problem, right? Because your mitochondria is not functioning properly, it is not producing ATP, and it is initiating immune reaction, and it breaks down lipids to produce energy, and that's generating a lot of free radicals, and your cell is under stress. And if you had healthy mitochondria, that would be the end of it. That would be the solution for many of your problems. Okay. Funny thing is, the mitochondrial problems are also present in Parkinson's disease patients, in ALS patients, in many of the neurodegenerative disease patients. The mitochondrial problems also exist. So imagine this: rather than having a drug for Alzheimer's, a drug for Parkinson's, a drug for this, a drug for that disease, how if we had a drug which improve, let's say, the health of the mitochondria? Then that drug may help some of ALS patients, some of HSP patients, some of Parkinson's patients, and so forth. So we don't need to have a definition of the disease. So then it's not important, and it's not relevant which disease you are characterized with. It doesn't matter. As long as we know what is the underlying cause for you to develop that disease, then it will be the root cause analysis, and we say, all right, we want to solve the problem at the causality level, right? So then there can be personalized medicine approaches, and drug companies would love that, because we can say, sir, you're not going to have a drug for this disease. You're going to have a drug for that mechanism, but your drug would also benefit Alzheimer's disease, Parkinson's disease, ALS disease, HSP and PLS disease, and many other rare disease patients. So, you broaden the spectrum. And that gives us an opportunity in 2020, in 21st century, to move the field forward so that we actually have more personalized medicine approaches. Because back in the day, when the diseases were identified and characterized, we did not know about the mechanisms, right? They were clinically identified, and by doctors, by clinicians, by the pathology in patients. But we didn't know at the cellular level, we didn't know at the molecular level, we didn't know at the genetic level what are the underlying causes are. But now we begin to realize that there is so much common between diseases and among diseases, right? So that we can actually focus our attention to those. So the rare disease factor, it may be seen as a limiting factor, but that's a huge advantage for us to develop personalized medicine approaches, and I think we have to build on that. So now, for the heterogeneity among the patient population, again, this may be a limiting factor, right, for some, because it is a limiting factor. It is that even though they have similar clinical outcome, the underlying pathology is not the same, which means one drug is not going to work for all. Then again, we can focus our attention to the common biology and common pathology, and then the definition of heterogeneity changes. We don't even need to consider heterogeneity anymore. We need to combine or group patients that develop the disease based on the common pathology. And then we'd say, okay, but then how do you know? Like, how can you find out, right? As long as we know the genes, the mechanisms, and also biomarkers, we will be able to group or subgroup patients based on the common biology and the pathology, the underlying cause. And I think that's very important. So now, the other limiting factor is the heterogeneity of the brain. Because if you look at the brain, it is extremely heterogeneous. And what do I mean by that? I mean that the cells and the neurons of the brain are very different. It's not like liver. It's not like your lungs. The brain has so many different cell types: neurons, astrocytes, macroglia. There is a huge variety among neurons: interneurons, excitotoneurons, neurons, projection neurons, local circuitry neurons, and even the projection neurons, thalamocortical neurons, corticospinal neurons, corticocortical neurons. The variety is immense, and they all live together. It's not that there's one region of the brain which is pure that neuron type. No, that's not the case. Every different neuron lives together in harmony in our brain. But in diseases, not every neuron becomes vulnerable at the same time. So for example, in HSP and PLS, it is the upper motor neurons that become vulnerable first, right? And by trying to help them, right, we don't want to make other neurons or other non-neuronal cells diseased. So this limiting factor, the heterogeneity of the brain, gives us a huge advantage for building cell-based treatments. So that means when we treat a neuron, we just treat that neuron without affecting the health of other neurons and other cells. And yes, this may be hard, but it is not impossible. So I think by changing our perception, the limiting factors become a challenge, but become also the initial starting point for the advantages that we can build in the future. And that's exactly what we're doing in our lab. And let me see. I like this a lot because, you know, it all depends on how we look at it. We can say I can, we can say I cannot, or I can't. I am with the vision of, you know, cutting the T out so that I can't turns into I can. So, you know, when I was a child, you know, many people told me, because I was short, that I could not be in the basketball team. Haha. So I was actually in the basketball team with this height. And then, you know, when I was born, they hold me from my shoulder and they injured my nerves. So I can't really lift my right arm, and I can't use my right arm as effectively as I can use my left arm. So when our school team for the handball school team was building, and, you know, people told me, you know, you can't be in the handball team, like, you know, in Soviet, right? I was in the handball team. You know why? Because I realized that not many people were left-handed and the left corner was always empty. So I scored a goal always on the left-hand side. So there's always a way, and it's in your brain. The moment you say I can't, you will not be able to do it. But the moment you say I can, then your brain will try to find ways of how you can. And I think we can. And that's why I actually never give up, and I work very hard because I think we can. So how can we, right? What is the way forward? And I think the way forward is teamwork, 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 collaboration, collaboration, collaboration. Because for complex diseases, it's very hard for one person to have all the knowledge, one person to have all the techniques, one person, you know, to do everything. Even in our brain, like, neurons don't work in isolation. They have to be in teams, they have to be in networks, and they have to work in harmony and in collaboration with other neurons and even non-neuronal cells. And it is the circuitry that makes things work, right? And I think for the scientists, we have to increase the chance of collaboration among them. So the layout or the setup is such that the scientists are actually forced to work alone, because the grant that they can get are personal grants. You know, they have their own lab, and everybody works in isolation, and that has been the setup for scientific advancements for, you know, for the past maybe 60, 70 years. But I think that's changing. So for example, in the left-hand side here, this is the ocean, big ocean, right? And of course, there are fish in the ocean, right? But because the ocean is so big, the chance for you to meet with another fish is not so high. But if you put all the fish in one pool, then the chance of those fish swimming together increases. And I think if we can put people together so that they have an environment where they can interact with each other, we can also increase the chance of collaboration among scientists. So that's why we want to initiate the Upper Motor Neuron Initiative. Our goal is to bring the scientists together, scientists who are working on topics that are related to upper motor neuron biology, to neurodegeneration, to drug discovery, to biomarker discovery, and for building cell-based therapies for the future. And then we have to create a collaborative and supportive environment. And to do that, you have to build trust. You have to build trust among scientists so they will know that when you give them something, it stays with them and you, and that there is a trust among scientists and they work together, and then they are trusted and they are respected and they are a team member. And we have to support the early stages of discoveries, because every tree was once a little shrub, right? It was very small. So every big idea or everything that was big was once small. So we have to support the early stages of discoveries. Then we also have to set the stage for translational medicine, because it's not just knowledge. From that knowledge, how do you translate to solutions? And there must be a path forward going from knowledge to solutions. And I think the solutions will be more personalized. And we're not going to have one drug that cures all. We're going to be developing personalized medicine options. And those options will lead to long-term treatments for patients. And I think that this is the way forward, and this is an attainable goal. And I think the time is now, because the patients have been waiting for a long time, and there is a huge development in the field of science and technology. And so we have all the ingredients ready, and the time is now. And especially COVID made us realize that one for all for one. Like, we make sacrifices, we stay home, and we give up from our freedom of going outside and hanging out and socialize, blah, 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 so that people that we don't know don't get sick. So that, people, we care for each other, and this is one for all for one. And I think time is now, so that we, as scientists and patients and drug discovery companies and everyone, get together so that problems that affect patients and humanity are resolved. And we are working very hard to make that happen. And I think we should form teams with patients and also foundations to move the field forward. So this will be my presentation for now, and I am very happy to take any question that you have. So thank you so much.